Hi guys, so we've had some practice on protein synthesis, and now we're going to talk about how changing the DNA sequence can have a big effect on the proteins that are made. So you'll remember that when we change a DNA sequence, that's called a mutation. So today we're going to look at some different types of mutations and how they can affect protein synthesis. So there's two main types of mutations. First of all, we have gene mutations, and remember, a gene is a section of DNA that codes for a protein. So gene mutations are within that gene changing a couple bases, or changing one base or multiple bases. Some of the examples of gene mutations are point mutation, frameshift mutation, and the different types of frameshift mutations are deletions and insertions. We're going to talk more about these in just a minute. There's also chromosomal mutations, which are kind of bigger changes where a chunk of a chromosome or even an entire chromosome gets changed. So with those, you can have chunks of chromosome that get inserted or deleted or switched around, inverted, or even moved from one chromosome to another in a translocation. And we talked about when we did meiosis about non-disjunction, where you can end up having when the meiosis messes up, you can have a whole extra chromosome or be missing a chromosome. So those are mutations that happen in big chunks of chromosomes or the entire chromosome. We're going to focus today on these gene mutations and their direct effects on protein synthesis. But it is important to understand that you can have chromosomal mutations also. So the first gene mutation we're going to talk about is a point mutation. And this is when a single base is replaced by a different base. So remember all the A's, C's, T's, and G's in the DNA? When one of those letters is switched to a different letter, that's a point mutation. And there's a couple different things that can result from this. So let's look at this graphic where we have our original DNA is TTC, so thymine, cytosine. What if we change that cytosine, the last base in the triplet, to a T? Well, when we go to the mRNA, that's going to be changed also. But then when we take the mRNA and look at our codon chart, both AAG and AAA are coding for the same amino acid. So even though we mutated one of the letters, we end up with the same amino acid. So the protein that's made will be no different. And we call that a silent mutation because it's not making any difference. Now, if we change the first letter of the triplet, and we end up having a U in the mRNA instead of an A, that's coding not for an amino acid, but that's a stop codon. And so the protein stops being made altogether. So that's making a really big difference in the protein synthesis. And then we can also have, so that's called a nonsense mutation, when you have a premature stop codon. A missense mutation is when you change a base and it ends up still coding for an amino acid, but it's a different amino acid than the original one. So there's multiple ways that can happen. And you see that when you change the last base in the triplet, there's a higher chance that you'll end up with a silent mutation. Whereas when you change the first or second base, there's not much chance that you're going to get a silent mutation. Now, frameshift mutations are, instead of changing one base, bases are going to be either added or deleted. And so when you do that, everything after the mutation is going to be shifted. So if you look at this graphic, and this is our original DNA, and right here, this is an insertion. So we added in this A, the adenine, and what it does is shift everything after it down. And so the amino acid that was coded for by the codon where the mutation happened, that one changed, but then also the next amino acid was changed, and then you end up having this extra letter hanging off here also. So that's a much bigger change than you might get in just a point mutation. And then down here, this is a deletion where this cytosine is deleted and it ends up shifting everything after it back. And so when we shift these letters back, we end up having UAG here, which you might recognize again, is a stop codon. So that's a premature stop codon, and that's a big change to the protein. So let's just revisit why mutations are so important to protein synthesis. So protein function is dependent on the exact structure of the protein. And that structure of the protein is determined by how it's folded. So remember that protein folding depends on the sequence of amino acids. So some amino acids like water, some amino acids don't like water, some of them like to bind with each other. So it depends on that sequence of amino acids and how they interact with their environment and with each other. That depends on how, that's what determines how the protein's folded. And then the way that the protein works is totally dependent on how it's folded. So if you change the sequence of amino acids, then the function of the protein can be totally affected. And this is an example down here. Hemoglobin is the protein that we have in our red blood cells that carries oxygen through our bloodstream. And so normally hemoglobin molecules just kind of float around in our blood. And if you change one base, if there's one point mutation, remember a point mutation is when you change a base from one letter to a different letter, you end up with mutated hemoglobin. And the mutated hemoglobin folds the protein's going to fold differently if you have that point mutation in the hemoglobin gene. And when the protein folds differently, instead of the hemoglobin molecules being able to kind of move freely, they clump up together. And that changes the actual shape of the red blood cells, and it affects the way that the red blood cells can carry oxygen and the way that they move through our bloodstream. We're going to look at that more tomorrow. So tomorrow in class, we're going to do some more work with mutations and look specifically at this hemoglobin example. So have a good night. See you tomorrow.